This is the first lesson of Chapter 11, where we're going to be looking into Mendelian genetics. Let's start off with a basic definition of genetics, topic that we're going to spend not just this chapter, but the next chapter as well when we look at molecular genetics. So genetics is the study of heredity and variation in living things. And you should recall from earlier parts of this class that DNA, as shown here, is the molecule that contains the hereditary information. It encodes for genes, and when condensed, that DNA forms chromosomes. And this should look familiar as well from our chapter on mitosis and meiosis. So we've been kind of setting ourselves up to learn about these more details of genetics in terms of how these chromosomes can pass on genetic information from parent to offspring, and that's where we'll go today. Let's start off with a little bit of the history for how we came to know what we do know about the role of DNA in genetics, and that was the first work was done by Gregor Mendel. He is considered the founder of modern genetics. And perhaps you've heard his name. He's pretty famous in the scientific community. And maybe you've heard of what his study subject was, what kind of organism he gained this information from. And those are the pea plants. Just everyday garden peas. You could even grow them at home. I've tried myself in Florida to grow them, and it's somewhat successful. You just have to battle with the iguanas, of course. But he was actually a monk, but he had an interest in botany and science, of course, having a lot of questions, and he wanted to explore those questions. So at the monastery where he lived, he was working in the garden. And he made some observations about the pea plants that they were growing that he decided to investigate through his research. But there were other plants that he could have chosen to work with. Why pea plants? So there's three main reasons why studying pea plants was Mendel's choice. First, and probably best reason, is that they're easy to grow. Also, there were commercially available strains. He could go to the local farmer's market at the time and purchase more pea plants as he needed them. Also, he was able to get what are called true or pure breeding strains. This has to do with the composition of the genetic information, and we'll dig more into that idea a little while later in this lesson. And there is another reason why that I just remembered, and that is they have several easily observed traits that are inherited, encoded by their DNA. So let's take a look at some of those traits now, because these are the characteristics that he was looking at when he was reproducing these plants and understanding how the pattern of inheritance happened in them. So here's a few examples of those inherited traits, such as the shapes of the seeds, which could be either round or wrinkled. The cotyledon color, which is the seed coat, the covering around the seed is the cotyledon, and it either was yellow or green. The color of the flowers were either violet or white. In addition, the pods, which contained the seeds, were either inflated or constricted, green or yellow, and their locations on the stems were either axial, which means along the stem, or terminal at the tops of the stems. So pea plants are a type of flowering plant, and flowering plants produce, when fertilized, when there is a successful fertilization between a male and a female plant, this is where the fruits form, and the fruits either are or contain the seeds for the plant to grow a new plant. So to understand how Mendel manipulated these crosses of pea plants, he basically wanted to control who, like, the mother and the father of the pea plant mating was. In order to do that, he had to manipulate the flowers. The flowers are the reproductive organs of the plants, flowering plants, that is. So we're going to take a quick look at how to identify the male and the female parts of a flowering plant. and then look at what Mendel actually did to manipulate these crosses. So as shown here, this is an example of a flower that has both male and female reproductive parts, but there are some flowers that only have male parts and some that only have female. However, this is not the case for the pea plant flowers. Pea plant flowers have both. So on the left, I'm emphasizing the male parts. Collectively, it's referred to as the stamen, and the stamen includes the anthers, which sit on top of the filaments. Basically, the filament is just a stalk that kind of props up the anthers. And the anthers are where the pollen grains are that contain the sperm. On the right side, we're showing the carpel. The carpel is the entire female reproductive structure, which at the top is the stigma. It's like a funnel that goes down to the style, which is an opening. And at the base of that is the ovary containing ovules or eggs. And if pollen containing sperm is able to travel down to the base, then it would potentially fertilize the ovule or eggs and result in a zygote, which ultimately would lead to the production of a fruit and seeds for a new plant. So for Mendel to control the reproductive crossings between individual pea plants, he had to manipulate their flowers. Let's say he wanted to cross a white flowered pea plant with a purple flowered pea plant. In this scenario, we're going to say that the white flowered pea plant is going to be the female in the cross, donating the being the one where the fruit will develop from a fertilized egg, and the purple flower plant is going to represent the male, or basically the sperm donor in this scenario. So the first thing that he needed to do is to remove the anthers from the flower that he wanted to serve as the female. And the reason for this is to prevent it from pollinating itself. So a little snip, snip. And now the white flower plant only has the ability to receive pollen from another plant. So it will serve again as the female. But he wanted to control this reproductive event. He wanted to make sure that the plant he wanted to provide the sperm is the one that indeed provide the sperm. So then he took a dry brush and dusted the anthers of the purple flower plant to gather the pollen, and then simply transfer it to the stigma of the female plant. And then wait and hope for successful fertilization. Of course, it's just a waiting game at that point to see whether or not it's successful. Then the emergence of the pea pod would be where the flower was, as you can see on the left here, where the flower petals are all kind of crumpled. This was a successful fertilization. So the pod contains the seeds or peas, and each of these peas has the potential to become a full plant, an adult plant once planted with the full genome for the pea plant. And they may also be genetically different. Each of these is a different offspring with their own set of DNA from the parents, and they could be genetically different or they could be similar. It depends on the cross of the parents. So let's go back to the original cross that we're following through here to see the emergence of these new seedlings from the parents that started with the purple flower and the white flower. And these are the actual results from Mendel's first round of experiments. And it was really interesting what he noticed. All of the offspring from that cross, or F1 generation, which stands for first filial generation, they all had only purple flower color. Not a single one had white flowers. And this struck Mendel as very interesting, and so he tried to find an explanation for that. We're going to be looking into this in the next lesson. So for here, I'm going to actually leave you with this idea of what Mendel's first experiment looked like. Now you have an understanding of how he could manipulate the cross to have a particular outcome. And we'll further explore this, do some Punnett squares, and then see how these experiments ultimately led to Mendel's laws.